Hi guys, my name is Tin and I works in the Nelson lab with Emily and we study the functional characterization of SLAC1 knockout in cotton. So basically to understand what we did, you have to understand the stomata. So the stomata is located on the underside of the leaf and it regulates the gas exchange and water loss within the plant. And within the stomata, there's this protein called channel associated 1. And basically this facilitates the release of these anions and it triggers the stomata closure. So some background is my mentor was doing this research before I was, and she conducted a RNA-seq where she did a differential gene expression analysis. And in this volcano plot, there is the downregulated and upregulated. And in the upregulated side, we noticed that there were two classes of biosynthase. There was starch and antioxidants. And so we wanted to replicate this experiment and measure these quantities. So we conducted a water limiting experiment. And in our water limiting experiment, we wanted to see how cotton responded to drought stress via physical and molecular changes. And we used physiological and biochemical assays. So in our physiological data, we use instruments such as the LI 6800 and FluorPen and others to create these graphs. So in these graphs, these are across soil water content, basically meaning how much water is in the soil. And this one is GSW, which is stomatoconductance, which basically means how open it is. The higher it is, the more open it is. And then WUE, which is water use efficiency. And so as you can see here, the green is the SLAC1. And as the soil water content decreases, SLAC1 stays consistently open here, and the water use efficiency stays consistently low. But we can see that wild type actually adapts to this, and it decreases its stomata, and it increases its water use efficiency. And so basically our main finding is SLAC1 is not adapting as well to the drought stress. So moving on to our biochemical responses, we did a starch assay, and that was to measure the glucose and starch accumulation. And then we did a DPPH assay, which was to measure the antioxidant activities. So from the starch assays, we collected these data using a spectrophotometer, and this is across soil water content, and this is the glucose activity. So as you can see here, there wasn't a significant difference between the two as time goes on, but there is a trend. As the drought stress gets worse, you can see that there's an increase in the glucose, which is what we biologically expected because starch is a necessary stable energy that they need to deal with these environmental stressors. Moving on to the antioxidant activity, this is percent scavenging across soil water content. And from the DPPH assay, we noticed that there is a significant difference between SLAC1 and wild type. And this indicates to us that SLAC1 is dealing with the oxidative stress and producing more biosynthase antioxidants to deal with the stress. And so concluding what we found, in the physiological data, we demonstrated that SLAC1 doesn't adapt as well as wild type. And then in the biochemical assays, we noticed that there was a trend in the starch accumulation. And in the antioxidant activities, we have to further study what is happening in this because it was a broad essay, because we didn't just focus on just one antioxidant. And hopefully our findings can contribute to developing a more resilient crop to drought stress. Thank you.
Great, thank you so much. This is super interesting and such an important question to help crops adapt to drought stress. I was wondering if you could tell me a little bit more, if you could continue with this research, what kind of research questions would you ask? Like what would you explore beyond this project?
So I feel like if we continue to this research, so what we did was very broad, as I kept explaining, because I didn't want to go on for too long. I think we would look into the antioxidants expressions more and use a higher sensitivity machine. So, because in our graph, we had a lot of variation, and that could be because of many reasons. But I think like if we had more time, we could look more into it and have more like data of understanding what antioxidants matter.
Yeah, sounds like you go in lots of different directions with this. Yeah, all really interesting. Great. Thank you so much for sharing your research with us. Yeah, this is great. Thank you.